So, hi everyone, I'm Lois, a clinical lecturer and PhD student from the Chinese University of Hong Kong. So, I would like to thank the organizer for giving me the opportunity to present today. So, a major research focus of our group would be liquid biopsy based on plasma DNA in our blood. So, it has wide application in both prenatal and cancer testing. But then, in our previous studies, it is mostly focused on the Illumina sequencing platform, which can only sequence molecules that are shorter than 600 base pair. And we didn't know whether there is an unexplored population of long cell-free DNA in plasma. Until 3 years ago, we have decided to use the long-read sequencing platforms, including the PacBio Smart sequencing and the nanopore sequencing. So we showed that actually in the plasma, there is population of long DNA up to tens of kilobases of length. And we have demonstrated the potential application in prenatal and cancer testing. But then, while we are seeing an increasing interest in our field about long cell-free DNA, we still know very little about their biological properties. So in this study, we reanalyzed our previously published data from healthy subjects, hepatitis B virus carriers, and also patients with hepatocellular carcinoma, which is a type of liver cancer called HCC, and also from pregnant women. And we recruited some new subjects and analyzed their plasma DNA with PacBio and nanopore sequencing. And then we studied the genomic representation, the molecular abundance, and the cleavage profile, that is how it is cut surrounding the CPG sites. So here, in this figure, the blue peaks shows the overrepresentation of long DNA, while the red peaks shows the underrepresentation. The darker regions in the horizontal track between the blue and red peaks represent the heterochromatic regions, while the lighter bands represent the euchromatic regions. So these are more transcriptionally active. And we can see that the blue peaks actually overlaps with the lighter bands, showing that the long DNA, they're preferentially derived from euchromatic regions. And now we can see that actually there is a higher long cell-free DNA abundance over the housekeeping genes and lower abundance over the unexpressed genes. And when we rank the protein coding genes according to the expression levels from low to high, from EXP1 to EXP5, we can see that actually there is a stepwise increase in long DNA abundance with the gene expression level. So having studied the relationship of long DNA with gene expression, we wonder whether there might be any clinical application. So we can see that there is actually a higher long cell-free DNA abundance over the cancer-associated genes in the liver cancer patients. And an ROC curve gives an AUC of 0.9 in distinguishing the patients with and without cancer when the number of molecules is at least 1 million per sample. And next, we study the cleavage profile surrounding the CPG sites. So in our previous study using Illumina sequencing, we showed that actually at the CPG sites, which is position 0 here, there is a twofold higher cleavage proportion for the hypermethylated CPG sites, while at position -1, there is lower cleavage proportion for the hypermethylated CPG sites when compared to the hypomethylated ones. And when we look at the long-read sequencing data in our study, we found that actually the pattern in long DNA is different. There is a preferential cutting that is several nucleotides away from the CPG sites for the long molecules. But then when you look at the lower panel, the short molecules, it is similar to our previous finding. And we then studied the cleavage profile of our HCC cohort. And we found that actually there is a higher cleavage proportion over certain positions, including -4, -2, 1, and 4 in the non-HCC patients. So we then, based on this finding, we developed a metric called a cleavage ratio, and there is a significant difference in the cleavage ratios between the cancer and non-cancer patients. So the ROC curve gives an AUC of 0.87 when we consider both long and short molecules. And after that, we would like to know more about how these long cell-free DNA are generated. So we studied mouse models in which different nuclease genes have been knocked out. So in this figure, we can see that the black line is the wild type, while the red line is the DFFB knockout mice. And we can see that there is a consistently lower proportion of long DNA in those DFFB knockout mice. And the DFFB deletion actually caused a reduction of long molecules originating from the regions surrounding the transcriptional start sites. So in summary, we showed that the long DNA molecules, they're preferentially derived from the transcriptionally active regions of the genome. They show a distinctive cleavage profile, and their characteristic distribution in the genome is at least in part due to DFFB enzyme activity. So we have opened a new possibility for biomarker discovery. Thank you.